So I'm Mark McCarthy from Oxford, so I'd like to welcome everybody here, add to the words of welcome. I know people have come a long distance, and hopefully we're going to have a fascinating couple of days. We've got a session now where I think the four groups, including myself, are going to say something about particular disease areas and the steps that they've taken to realize vision and understand both variant discovery and function for those particular diseases. But maybe just you can click through that, just click. Okay. And I think this will also underpin some of the areas where there's clearly a need for further development. So I'm going to talk to you a little bit about some of the work that I've been doing and my team's been doing with collaborators here in Oxford and, of course, around the world to try and understand a little bit about type 2 diabetes. And to the extent that type 2 has been one of the leaders in the field in terms of prosecuting variant discovery and function, I think it comes down to a number of areas. One, the high prevalence of the disease means we benefit from large sample sizes. That's allowed us to get to large sample sizes for case control. And of course, as the biobanks and EHRs have become available, we can tap directly into those because of the high prevalence of disease. We're fortunate in having a lot of intermediate phenotypes that we've been able to use to understand the physiology of the hits. We have a certain amount of confidence about what the tissues are that matter for type 2 diabetes. And what's more, we have access to all of those tissues to variable extent in humans and good cellular models for many of them. And I think particularly important, we've had a very strong collaborative history, particularly in the genetic space, increasingly now in the functional space, a lot of pre-competitive work with pharma and a lot of support from major funding agencies. So the way, probably over the last 5 years, we've approached pinning all these pieces of data together is the somewhat linear approach here that I'll suggest we've moved beyond by the end of this talk. But the idea that we get genetic signals, we're clearly particularly interested in identifying the genes through which they operate, the functional candidates, and then stitching those together into pathways and networks we can get from the signals to the genes, sometimes through fine mapping, identifying the causal variants. And if they're coding, that points to the genes. If not, we have to work out what the regulatory consequences of those variants are. But sometimes we can get to it straight through understanding of what the genes are in the region, the ones that are the best biological candidates. So just to take you through some of those steps, in terms of detection of association signals, we've been able to amass large data sets, particularly in the GWAS space. We recently published on about 900,000 individuals. We've now got a larger transethnic GWAS in somewhat over 1.25 million individuals, 500 loci appearing from that. We've explored the rare variant space as far as possible with exomes particularly, and starting to dip into the genome space, but clearly those remain substantially underpowered, particularly to detect things we haven't seen in the much larger GWAS. But that's giving us clear insights into genetic architecture, polygenic risk scores, ability to do Mendelian randomization experiments, for example, to characterize potentially modifiable non-genetic risk factors. And the things we need to prosecute that going forward are ones that are going to be generic to many diseases. It's really about, since finding risk alleles is the substrate through which human genetics operates, we want to maximize the number and the diversity of those as far as possible, using a variety of approaches that will maximize sample size and minimize friction in bringing those data together. When it comes to moving from signals to variants, I think we've been quite surprised at how effective fine mapping has been as we've built the sample sizes up and been able to exploit better reference panels such as the HRC. In the latest transethnic meta-analysis now, we have about 150 signals which we got down to single base pair resolution. In other words, a posterior probability of association of greater than 75%. So that's crucial in terms, again, of understanding genetic architecture, absolutely vital to understand regulatory overlaps and start to learn the rules of regulatory grammar for gene-gene interaction studies, of course, and for defining the effect of transcripts. If you see a coding variant signal that's associated, it's tempting to think that that points directly to the gene, but unless you've actually explored the local LD architecture, you could be confusing yourselves because it could still be that it's just hitchhiking on the background of a regulatory variant. The fine mapping will only get us so far. In many loci, we can't deconstruct it, and there we have ancillary approaches, which are rather more tissue aware than the tissue-agnostic approach we get through genetics, mapping regulatory annotations in the tissues we care about, developing high-throughput variant assays, exploiting machine learning as ancillary tools to understand which of the variants in our regions are likely to be driving the signals. And again, I return to the point I made before, we're quite fortunate in that we have a pretty good sense of what the key tissues are likely to be for type 2 diabetes. Some of them are already well covered in GTEx and ENCODE and roadmap. Others, like the pancreatic islets, have not. And we've had a concerted effort here with Anna Gloin, but many other people around the globe, some of whom in this room, to really understand the regulatory apparatus of the islet. Key, because everything we see from the physiology, everything we see from the epigenomic evidence is that the islets are the main tissue of interest. We see much more signal from islets than we do, for example, from muscle, where we see very little signal, with liver and adipose tissue somewhere in between. I think a really key point that we've appreciated recently is that to actually understand and exploit the functional evidence to its full and to think about appropriate functional follow-up stimulants, we need to understand at any given locus what the tissue of action is. So this is just some work that a postdoc in my group, Jason Torres, has been doing to bring together genetic fine mapping data, patterns of genome-wide annotation overlap, the specific overlaps you see at individual loci, to apportion those 400 plus loci we've got into those that we think are working through the islet, those that are working through fat and muscle and so on. Absolutely vital, because, just to be crude, if your region is clearly operating through the islets, you don't want to be investing too much energy in interpreting a liver screen or a liver EQTL, because it's probably irrelevant. So the things we need in that space are really to elaborate that catalogue of regulatory consequences of genomic variation. I won't go through all the points there, but we're only scratching the surface in looking at bulk tissues, in mature tissues, in basal states. We need to be stimulated, looking through development, single cell, and all the rest of it, for a wide variety of assays. Probably the key area where we've put most energy over the last 2 or 3 years is to think particularly about the effector genes. After all, this is our connection to biology. After all, it's the point of interest of most pharma companies is to identify the effector transcripts that are the targets of these genetic variations. A variety of different approaches we can bring to bear there: genetics, biological assays of gene function, our ability to take the regulatory signals and map them to genes and perturb them. Essential for getting to mechanisms and direction of action, a clue towards validated therapeutic targets. And now I think a key thing we need to do is, again, build up the catalogue of those relationships with a variety of different methods. I think we're all conscious that we exploit cis-EQTL data or high-C data. We can tell nice stories about how those connect our regulatory variants to the genes that we think might be targets. But that of itself doesn't demonstrate that those genes are on the causal pathway to disease. I think we're all aware of that these methods have limited specificity and sensitivity, and actually it's a coalescence of different types of evidence that will give us believable data. Sometimes it works. Here's an ADSI3 locus, which many of us in the diabetes field put up as one of our best examples, where we know the associated phenotype, we know the causal variant, we know the tissue of action, we know the effector transcript, we know the direction of effect. Tempting to rest on our laurels there and say, this is done, tick that off, don't look anymore. But Melina's got some very nice data suggesting there's more going on in this locus than this simplistic, deterministic kind of assessment. So I think one of the key questions that we need to be thinking about in a field like this is what are our standards of evidence? What are our standards of proof? When do we stop? When should we reevaluate? In type 2 diabetes now, we've got about 60 gene targets for which we've got either very strong or pretty strong evidence that these are genes through which at least part of the type 2 diabetes effect is being mediated. These are potential therapeutic targets, clues to new biology, but they are really only hypotheses until we've really, as Eric said, wrestled them to the ground, something we and others have been doing at the single gene level. We absolutely need to scale up to do this at scale using a range of cellular and in vivo models, and then knit all this together into networks and so on. Work that Anna Gloin, who's maybe here yet, I'm not sure if she's here yet, but has been doing in Oxford, performing screens, knockdown screens, initially with shRNA for a limited set of genes in in a human pancreatic beta cell line, and then taking those now to pooled CRISPR genome-wide screens, loss of function, gain of function, looking at a variety of cellular phenotypes, giving us additional data that we can bolt on top of those hypotheses. This is clearly an area where there's a huge amount of work going on and a huge amount of work that needs to be done to think about those cellular assays, to develop better cellular models, probably iPSC derived, to think about better readouts, ones that are particularly relevant to disease, of course, and a range of better in vivo models. And then let me just finish off with just one word on translation. One of the really gratifying things to see as we've built up these GWAS studies to bigger and bigger scale is our ability both to detect really, to generate really good overall polygenic risk scores, and we can argue about how those are going to be played out in terms of their ability to predict risk. In the case of type 2 diabetes, you could argue that we can do quite a good job with measuring scales and a calendar and a glucometer, so you might argue how much genetics is going to add to that. One area where we think genetics will be absolutely pivotal is in relation to understanding the heterogeneity within type 2 diabetes. So the ability to deconstruct that overall risk score into the portions of risk that are operating through the beta cell, the portions that are working through adiposity, the portions that are working through insulin action, will be, I think, a very important tool for understanding difficult clinical questions about what we do when somebody's diagnosed with respect to complication risk and therapeutic options. One area we've put a lot of work in, you can see that was a screenshot where I was obviously, is to make as much of our data available as possible. So we work very closely with folks at the Broad and elsewhere to develop this portal where as much of the data, both genetic and genomic, is readily available to the community. And then this is really my last slide, which is to say, really, I think building on some of the points that Eric made, but perhaps conceptualizing it slightly differently, we've certainly seen this over the last few years as a linear process of variants to genes to pathways. But as we build more and more genome scale data in all of these areas, it becomes more of a virtuous cycle where we feed in lots of different data relevant to a given locus from the genetic, from the functional perspective, from the clinical perspective. And when we reach some kind of level of crescendo of coalescence of information, we can tick that off as an established mechanism that obviously then feeds back as priors into understanding what's going on at some of the other loci. When will we stop? I guess at the point at which the biological model is saturated. If we keep generating, hitting the same bits of biology time and time again as we go through the cycle, I think it's fair to say that we'll have reached some point of coalescence. So let me finish there. Many people I could mention. I won't go through them all by names, but I'm very happy to take any questions. Thanks.
Mark?
Yeah. Oh, yeah, there you go. Sorry. Hi, you.
Okay.
How did I know it was going to be you asking the first question here? Stereotype.
I'd hate to break a tradition. That was a lovely talk. I was curious about those loci that you kind of got down to a gene.
Yeah.
And a sort of naive view of human genetics is that, as you say, these things should converge. So eventually, the kind of variant signal in the sort of non-synonymous space or stop codons should agree with any kind of regulatory link that links you to that. But of course, not all of your things said coding variant or RVAS or whatever it is. To what extent do you think that slightly perhaps naive view is playing out? That is that every gene that, I mean, the naive view would be that every gene that is a true player will have, at some allele frequency, a non-synonymous variant that is a risk allele.
I mean, I think with 7 billion people sequenced, we probably find that was true most of the time. I mean, not always, of course. And I think a really key point, which is I think underappreciated, is that the phenotypic spectrum of the regulatory GWAS signal and that of a coding signal may not be the same. And we tend to think they equilibrate, but they're as dissimilar as a global knockout and a tissue-specific knockout. They could have very different phenotypes.
So I want to take issue with Ewan's question. I don't buy that at all. If I have a bunch of enhancers, some of which are tissue-specific enhancers, that will increase the amount of a protein in a given tissue, I see no reason why there should exist a coding variant that will accomplish the same thing. So it's a hypothesis you're raising, but I think we should expect that changing levels of a protein are not the same as changing the protein. So who knows
Yeah.
is the answer.
Yeah.
I mean, I get that. I think the naive, well, anyway, I think it's interesting to really get data on this directly. To what extent does one in practice see a kind of mimic, not mimic, but a complement on the non.
Yeah, no, I mean, I take that point. I mean, we've got lots of examples. I mean, we could quote you where we've tried to do those experiments where we have seen cognate rare variants, and Anna's reported on several of these, cognate rare variants in GWAS signals. And, you know, the phenotypic spectrum is not the same for the very simple reason that the tissue, the coding effect will be in many more tissues than the regulatory GWAS effect by and large. And therefore might have a more pernicious effect that doesn't even lead to, survival. Yeah.
I was just going to make a general point that since we've got a lot of example talks in the morning, that maybe this is a great topic for discussion, but maybe we want to go through the individual examples and then take this up in the group discussion.
Yeah, no, that's good.
General issues.
Okay, let's do that. Anyway, my 15 minutes is up anyway, so.